Hello, everyone. Thanks for tuning in to this presentation. My name is Nathan Snow. I'm from the USDA National Wildlife Research Center. We'll be talking today about field evaluations of sodium nitrite toxic bait for wild pigs. And I'd like to start out by acknowledging my co-authors, Jason Wishart, Justin Foster, Linton Staples, and Kurt VerCauteren. As you can tell by all these pictures, this has been a large undertaking and an international collaboration to get to where we are today. From the USDA, it's been the National Wildlife Research Center, the National Feral Swine Program has provided some funding, and the Wildlife Services Operations has been really helpful in getting us access to properties and helping us out. Some external collaborators are the Texas Department of Parks and Wildlife, Texas A&M University Kingsville. We've received some funding from APHIS. And then internationally, we work with Animal Control Technologies Australia. They are the bait manufacturer, and we've been working really close with them. It's a picture of their facility here on the screen. The Invasive Animals Cooperative Research Center in Australia, and then Connovation in New Zealand. And so I'm going to give a little background before I get into some of the field results. So why are we working, where did this all start? And it's this publication right here. Some Australian researchers trialed some potential toxicants in 2008, and sodium nitrite came out on top as the best new potential one to go forward with. They approached the USDA, and our leadership agreed that we would try to pursue this as a toxicant for registration in the United States. And so why did we like sodium nitrite? First, it's because it's acutely toxic, meaning pigs typically die within 1 to 3 hours, and they're not found very far away from the bait site. It's humane death. The mode of action is similar to carbon monoxide poisoning, and the mode of action is actually called methemoglobinemia, which is reduced ability of the red blood cell to carry oxygen to the tissues. It's a food preservative, which is helpful for registration because we already know quite a lot about sodium nitrite. It's in a lot of the preserved meats that we eat every day. And so there's quite a lot of research already done. And then originally, back in 2008, we thought that pigs were very highly vulnerable to sodium nitrite, more so than other species. And recently, it's coming to light that we're finding out that that's not really true. Most species are pretty sensitive to sodium nitrite. And so just to give a little more information on that point, here in this graph, we have all the species that we have tested with sodium nitrite. And on the y-axis is their LD50, which is basically a level of sensitivity of each of these species to sodium nitrite. And LD50 would mean that it's the amount that an animal would have to eat that would kill 50% of the animals that eat it. And so you can see that a pig is basically smack dab in the middle of all these other species. So there are some species that are more sensitive and some that are less to sodium nitrite. And the tough thing that we're dealing with here is that a pig needs to eat about a golf ball to baseball size amount of this bait in order to get a lethal dose, whereas something like a small bird needs to eat a speck of this stuff. So the smallest little fragment of bait here is 0.0 grams. They don't even need to eat that much, like a small passerine bird, to get a lethal dose. So here's a timeline of the progress that's been made on this bait. As I mentioned, in 2008, that Achilles heel paper was published. From 2009 until 2012, there was some testing of some prototype baits here in the US. Pretty much all of this was non-toxic, and anything that did include sodium nitrite, that we weren't having any luck getting pigs to eat it. In 2013, we conducted some of our first pen studies with sodium nitrite baits. And we realized that palatability of this stuff is really a big issue. Sodium nitrite is basically a salt, and so it's in really high levels in any sort of food. It's unpalatable. So we had to figure out a way to hide it. And in 2014, we figured that out with some microencapsulation coating. And this is the first time that we actually had good success with getting pigs to eat this bait reliably. In 2015, we worked on making a bait station and baiting strategies to make sure the bait station is to make sure that no non-target animals are going to be able to access the bait. And then we need a strategy basically to teach pigs how to use the bait station before we can deliver the toxic bait to them. In 2016, we did some pen trials that were submitted to the EPA. This goes toward registration. And we achieved 95% mortality in the pens, which exceeded the EPA standards that we needed to. In 2017, we looked at humaneness and secondary hazards and did some biomarker studies with placebo baits. And we found out that the secondary hazards are very low to any non-target animals, and that it's a humane bait. In 2018, this is basically where the rest of this presentation is going to start from. We did our first field trial in Texas, USA. We found pretty good efficacy for pigs, but we found some non-target issues with that bait. And so then in 2019, we reformulated the bait to try to reduce some of those non-target hazards. And we conducted some field trials in Queensland, Australia first, and then in Alabama, USA, and then more recently, just finished some in Texas, USA again. And that's in 2020. That brings us to now. Okay, so I'll just give you a brief introduction to what our baiting strategy is. And this is basically what we followed for all of these studies. So on nights 1 through 6 is when we would just put corn out on the ground, and this was to try to locate pigs. And we would choose the best sites, meaning the bait sites that had the most pigs coming to them. On nights 7 through 8, we would introduce the bait station, and we would leave it open about 10 inches. So pigs would have to feel the lid on their head, but didn't have to do much effort to open it up. On night 9, we would close the bait station down, so there's about 2 inches. So they would have to lift the lid a little bit to get into it. On nights 10 through 11, really on nights 10 through 14, we locked the lids of the bait station with a magnet. So it takes about 30 pounds of pressure to lift the lid now. And so nights 10 through 11 are when we put placebo bait. So this is exactly the same as the toxic bait, just without the sodium nitrite. On nights 12, we put in the toxic bait. And then on nights 13 through 14, we put back in the placebo to look at how many pigs were still remaining in the area after toxic baiting. And so this is basically what we're looking for when we know we're ready to deploy toxic bait. These pigs just run in, open up the bait station, and start feeding all together. We really try to take advantage of this feeding frenzy that pigs seem to have when they're feeding together. I think it helps them consume enough of the toxic bait. So the first field trial, I'll go back to Texas in 2018. This is one that I presented at the last Wild Pig Conference. I'll just go over this briefly, what happened there. So the study area, we had about a 10,000 acre pasture in March 2018. So this was pre-green up, which we were hoping would be good for getting pigs on bait. We put 74 GPS or ear tag transmitters onto pigs, and we had 14 bait sites. The results from that are we had 66% knockdown in the population in just 1 to 2 nights of baiting. We found 109 pig carcasses, although we know there were more that we just couldn't locate. And the average distance that a pig was found from a bait site was 150 meters. So that was all looking good, except for this. We noticed that that pigs were spilling bait quite a lot. And you can probably see in this picture, that's a lot of spilled bait out there. If I would put all this together, it'd be probably about the size of a grapefruit. And so we thought maybe that the way we presented the bait, and that also maybe there's some palatability problems with the bait that led to them creating this huge mess here. It seemed like they were searching through the bait to try to find something that they wanted to eat. And so that dropped bait unfortunately led to quite a few non-target deaths. We found 164 passerine birds. Most of those were white-crowned sparrows. We found 4 wild turkeys and 8 raccoons. And I want to say it's important to know that none of those actually accessed the bait station. They all died from consuming bait that was spilled outside the bait station by pigs. And and I also put these pictures here just to show you that we were really testing this stuff. Well, our bait sites had tons of non-targets at them, and so we really want to know basically how this stuff is going to work if it ever gets registered. And so obviously that wasn't good enough for us, and so we made some modifications to the bait. After that study, we reformulated. I have 2 pictures here of the reformulated and then the old stuff. The reformulated bait, we took out 50% of the sodium nitrite, hoping that if anything does get spilled, there's going to be 50% less sodium nitrite on the landscape. We also enhanced the microencapsulation coating that protects the sodium nitrite in hopes that pigs would not detect the sodium nitrite and would eat it more readily instead of spilling it. And then we also replaced the cracked grains with grain flour. We did that because if any of this gets spilled, we felt that the cracked grains were probably pretty attractive, especially to some of those small bird species. And so we didn't want to have that laying on the landscape if any spillage occurs. And then we also modified the way that we are presenting the bait to pigs. And so on the left is the way that we did it in 2018, which led to a lot of spillage. We crumbled it and put it in the bait station, and we felt that that probably made it pretty easy for the pigs to spill. And so then we tried one method where we separated the bait station into troughs or into separate compartments, and we compacted the bait in by hand with hopes that basically a pig would only be able to take about a mouthful and walk away and eat it and not scoop any of this of the crumbled stuff out. And so and then the last method that we tried, which we thought was the best and the easiest actually for applicators, is to have the bait already put into a tray, and it's nice and compacted into that tray. And then all we need to do is slip the tray into the bait station. Okay, so after we've made all those modifications, we went to Queensland, Australia to test them out. And we went here because at this study area, we felt like there was very low issues with non-targets coming into the bait and eating it, just because they weren't really present in this area as much. And so if we were going to have another big spill, and we wanted it to be in an area where there just weren't very many non-targets. So so we went to this about 25,000 acre ranch in December of 2018. It's a time of year where it was really hot and dry there, so pigs were easy to find around the watering sources. We put out 15 toxic bait sites, and we did similar pre-baiting as I mentioned previously. We didn't have any GPS collars on pigs here. We just monitored pigs pre and post toxic baiting with cameras in order to get an estimate of mortality. So the results from this were, we found that we removed 90% of the pigs in one night of toxic baiting. And so compare that to what happened in Texas, which was 66%. So we increased our pig lethality. The average amount of dropped bait was 55 grams. So that's about a 19-fold decrease from what we experienced in Texas, which was an average of about 1 kilogram per bait site. So 55 grams, you can picture is um, smaller than a golf ball, basically. And then we did find 3 dead non-targets, which were all crows. In Texas, we found 167 dead non-targets. So non-target death went down, but as I mentioned, we purposefully went here because we expected there shouldn't be very many non-targets in the area. And all 3 of those crows, I'll say, also died from eating bait spilled by pigs. But we thought those results were quite promising. We removed a lot of pigs and not very many non-targets. So we felt it was safe enough to come back to the US to do some more trialing here. And so in 2019, in August of 2019, we went to Alabama, about a 4,400 acre hardwood swamp property in August when it was hot and humid. And so pigs congregated onto this property from surrounding areas to get to some of the water resources that were there. We had 10 bait sites. 5 of those were toxic and 5 were controlled. And control meaning we never deployed toxic bait. We just kept everything placebo at that site, at those 5 sites. We did similar pre-baiting, and again, we monitored with cameras to get measures of lethality. So the results from this are in one night of toxic baiting, we removed 78% of the pigs. And we actually know that we could have removed more pigs. It was surprising to us how many pigs were on this property. And you can see from this picture that these trays of toxic bait are entirely licked clean. So we didn't have quite enough bait out there to kill every pig. And so if we would have offered more bait, I think that that 78% would have been higher. But anyway, 78% is still really good in one night. Compare that to Texas, where we had 66, and then Queensland, Australia, where we had 90%. Dropped bait, we could hardly find any. And I think that might be because of having a shortage of bait out there, which actually might be a strategy for how we deploy this bait. I think pigs cleaned up after themselves quite well here because there wasn't quite enough bait. And so you can compare that to what we found in Texas, which was 1 kilogram of dropped bait at a bait site, and then in Queensland, 55 grams. And then we only found 2 dead non-targets in Alabama, and those were both possums. And actually, one of those couldn't be confirmed to be sodium nitrite killed. The other one definitely was. But compare that to Texas, where we found 176, and then Queensland, where we found 3. And so I just want to mention 2 things here, or actually just one thing. The fact that these pigs were licking these trays and cleaning them up so well means that I think that we really fixed some of the palatability issues that we were originally suspected were occurring with this bait. Okay, so then this brings us to the 4th field trial, which happened in Texas in 2020. It happened right before all the COVID outbreak stuff in March. And we wanted to go back to almost the same spot that we trialed in Texas in 2018. And so you can see in the little inset map of Texas here, the black dot is where the original trial, where the original trial took place. And the red dot is where this 2020 trial took place. And so we really wanted to be in a similar landscape with similar non-targets to see to get a real good idea to if we made any improvements or not. And so we had about a 13.5 our 13,500 acre pasture land to work in. Again, we went at the same time of year as in the 2018 study, so in March. We actually deployed toxic bait on the exact same day as we did in 2018, but in 2020. And we followed the same methods as we used in Alabama in 2019. And so the results from this are we found 81% reduction in pigs in one night of toxic baiting. And so compare that with some of the other studies. You can see that we're pretty high. We have pretty high lethality in all of these studies. But Queensland, Alabama, and the last Texas study all have the highest. Dropped bait, we did find some. We had about 30 grams at the most at one site to basically 0 at some other sites. So anywhere from 0 to 30 grams. Unfortunately, we still found some dead non-targets, and they were all these small birds. And we actually found some dead non-targets at some sites where we couldn't find any spilled bait. And so this just really goes to show how sensitive these birds are. The smallest speck of any spilled bait could kill a bird. And so we found 35, and they were all dark-eyed juncos or white-crowned sparrows. And so just some conclusions from all 4 of those things. I think that basically we're making some good improvements since the 2018 issues, but we're not quite there yet. We've improved bait consumption and lethality. We substantially reduced the amount of dropped bait. It's very, very minimal. If you can imagine a group of 12 pigs coming in and eating together and fighting each other, and only having 30 grams of dropped bait, I think is actually quite a success. But it's not enough to reduce non-target deaths. And so we still have some work to do. And I think the next step that needs to be done is basically we need to find a way to deter birds from the baited area, because these birds are really the species that seem to be the most vulnerable to any spilled bait. Even trace amounts of dropped bait will kill a bird. And so we're already working on that. Actually, we've been allowed to do some field work during all this COVID-19 stuff. We've been working around here in Fort Collins, Colorado, to figure out some ways that we can deter birds from bait sites. So basically, we're baiting in birds, and then we're trying to deter them from eating the bait that's at that bait site. And so some of the things that we're trying are a mesh grate, which we could put this bait station on top of a mesh grate, and any spilled bait would fall through that mesh and be unavailable to birds. Methylenthrenolate is a chemical that is known to, or has been researched and shown to be repellent to birds. And so we're trying to spray that on the ground around the bait site. And actually, methylenthrenolate, interestingly, might actually be attractive to pigs because it smells it smells like grapes. It's basically an artificial grape flavor. And then we're going to try some scare devices. And this study is currently underway. We're collecting data right now. That's a picture of us working in the field with our face masks on. And we just finished testing 3 of these things: the mesh grate, a scary man, and then the methylenthrenolate. And the mesh grate seemed to be basically the only thing that stopped birds from eating bait that you had them baited into. The scary man didn't seem to be very scary, and the methylenthrenolate might have worked for about 3 minutes, and then the birds were used to it and they kept eating. So we have a couple other things that we're still going to test, and then but the good news is that the mesh grate does seem to work. And so we at least have an option to move forward with. And we're going to ask the EPA for permission to conduct another trial down in Texas, hopefully in the same area that we were just working in. And we'll see if using this mesh grate underneath the bait station is enough to keep that bird mortality to 0. And we're hoping to get this study underway this summer and get some results so that we can continue on toward trying to register this product. And I'd be happy to take any questions if you guys want to email me or ask Jessica to email me. So thanks a lot for listening, and everyone stay safe.